you'd have to ask her the question distribution. She'll be assembling the test.
Okay, will do.
And so far in neuroanatomy, I have 30 questions, and I have more to go. So, again, you'll need to clarify that with her. So, here we go. We're going to start today with the basal ganglia, and then move into the thalamus, hypothalamus, and pituitary gland. When you talk about the basal ganglia, which consists of these deep brain nuclei here, the bottom line is, and I'll go over how in just a minute, but the bottom line is these guys control parts of movement. And they're not pyramidal cells, as you'd see in the corticospinal tract. They are extrapyramidal, and I mentioned that before. So when I talk about the extrapyramidal areas of brain, I'm talking, one, about the basal ganglia. Okay? If you look at this slide here, the basal ganglia, the caudate, this thing that you see right here is called the putamen. The putamen. On the inside of the putamen, on the medial side, is a structure called the globus pallidus. The caudate is another nucleus that is an extension of the putamen. So this would be the head of the caudate nucleus. And then this nuclear group goes up and around like that as the body of the caudate and the tail of the caudate, ending in the amygdala. Now, neuroanatomists have classified different groups of these deep brain nuclei. They go by different names. Well, one name that you see over and over is the striatum, which means the caudate and the putamen. In other words, this one right here, the caudate and putamen is the striatum. Now, medial to the basal ganglia, you'll see the thalamus here. And between the thalamus and the basal ganglia is a structure called the internal capsule, which is this right here. The massive fibers, this is the internal capsule. These are the axons traveling up to and out of the cerebral cortex, going down to the brainstem and spinal cord. So that's the structure. You've got basal ganglia out here, internal capsule, thalamus, and then next to the thalamus is going to be the third ventricle, and on the other side, you've got thalamus, internal capsule, basal ganglia. Okay? That's the scheme. If we take this section here through the brain, you're going to see where we are here. That section is far anterior, and you'll see it doesn't cut through the thalamus. So in this section, you don't see the thalamus. What you see here is the outside of what we were just looking at. So right there is where we were looking at. So we're, from the outside, there's the putamen right there. Inside of the putamen is the globus pallidus. Oh, the words are over here. Is the globus pallidus. Now understand that the caudate started at the head of the putamen. And as it comes up, it's sweeping up like that. It's sweeping up. So in this section, we've caught the caudate where it's sweeping up. See what I mean? And you can see it up there in that top margin. You can also catch a little bit of the amygdala out here in the temporal lobe. Now we're going to take the section back just a little bit, and if we take it back just a little bit, here's our putamen. There's the globus pallidus. There's the caudate up there. It's been sweeping back. There's the internal capsule. There's the thalamus. There's the third ventricle. Follow me? Here's the amygdala down here. You got to keep your frame of reference on this slide. All right, now we're going to take the section back just a little further. Oh, excuse me. We're going to do a coronal section. Let's do a transverse section here. View it in transverse section. Here's the putamen. There's the globus pallidus. Here's the internal capsule. There's the thalamus. There's the third ventricle. Now, look what happens to the caudate. The caudate came out, and in our section, we caught the front end of it, right there, and as it sweeps up and back, we caught it again back here. Look at where you are up there. You caught it in both places. Follow me? Understand that these are not fiber tracts. These are collections of cell lines. These are nuclei arranged in this shape and in this long horn-looking shape. One thing you should know is that the caudate forms the lateral wall of the lateral ventricles. Also, the thalamus, right here, forms the lateral walls of the third ventricle. Caudate forms the lateral wall of the lateral ventricles, thalamus, walls of the third ventricle. Afferents that project to the basal ganglia come from the cerebral cortex, all over the cerebral cortex. They send projections to the basal ganglia, and specifically to the striatum, to the caudate and putamen. That's kind of the receptive area of the basal ganglia, the caudate and putamen. You also get projections from the substantia nigra. We actually know that from yesterday's lecture. The substantia nigra projects its dopamine fibers to the caudate. Caudate projects back down through GABAergic fibers to the substantia nigra. You receive projections from the thalamus and from an area called the subthalamic nucleus. All of that afferent information going to the caudate and putamen primarily is relayed to the globus pallidus. And you know, I cannot remember what these stand for, so don't ask me that. I looked at that slide and I thought, what did I put that on there for? I cannot remember. But all information is relayed to the globus pallidus. The globus pallidus is the output of the basal ganglia. So the globus pallidus projects back to the thalamus, to the subthalamic nucleus, to the red nucleus. Now, one thing let me say about the thalamic projection is that the thalamus projects back to the cortex. So you got cortex projecting to the striatum projecting to the globus pallidus, the globus pallidus projecting back to the thalamus projecting back to the cortex. We talked earlier about the red nucleus, and if you cut the red nucleus above the red nucleus, you wind up with posturing. That's indicated there. This is one thing I want you to pay very much attention to, is that the output of the globus pallidus is all inhibitory. It's all inhibitory. This schematic here provides for you the afferent supplied to the basal ganglia and the efferent projections from the globus pallidus. It's just what I said in the previous couple of slides. So here are a couple of things you need to appreciate. Even though the basal ganglia is involved in movement or muscle function, there are no projections from the globus pallidus directly into the spinal cord. It exerts its influence by projecting to other things that project to the spinal cord, like the red nucleus, for example, or back to the cerebral cortex through the thalamus. The second bullet point, because we're talking about an inhibitory system, it provides these feedback loops to dampen movement. That third bullet point, you need to really appreciate it, it provides an intact basal ganglia, provides a tonic break on movement. Remember we were talking about you sitting there the other day and you're not moving, but you have muscle contractions? If we remove the brake, you release motor activity so that now you start with tremor. Different types of tremor. We went over the other day the difference between resting tremor, which is a function of the basal ganglia, Parkinson's disease, substantia nigra, projection to the caudate, versus movement or attention tremor with a lesion of the cerebellum. Athetosis is another type of tremor, and it's characterized by and I'll sort of demonstrate it here. If you've seen people, it's sort of a worm-like movement like this. You know, they usually have severe problems, but it's not a patterned movement like that. It's more of a, it says writhing, and that's what it looks like. Choreiform movements, like in Huntington's chorea, they're wild, jerky movements, like more like this, that they can't control. These are all described as dyskinesias. And they all are related to a release phenomenon, disinhibition, that I just described. You remove the GABA inhibitory input, you release the structure to fire away. Parkinson's disease we talked about yesterday, or sometime, with the different symptoms related to Parkinson's disease. One other type of dyskinesia that's interesting is called tardive dyskinesia. And you see this a lot in people who have Tourette syndrome, or it's usually a side effect of a drug. And what these are is they're rhythmic movements, particularly of the face, like, you know, they do like this, you know, and lip smacking movements, and they can't stop them. If you've ever seen a kid with, or a teenager with Tourette syndrome, It's very common in Tourette's. But that's called tardive dyskinesia. We don't use Haldol and Prolixin very much anymore. There are other drugs to treat the psychoses. But I'll tell you what, if you wind up in the emergency room and you have somebody having an acute psychotic reaction, if you want to mellow them out real quick, Haldol IV works real good. The dose is 1 to 5 milligrams. Give them the 5, and they will chill. Okay? Any questions about that?
So tardive dyskinesia, is that specifically to the face, or is it specifically to Tourette? Like, what's the definition of tardive
It's
dyskinesia?
tardive dyskinesia. It's rhythmic movements, particularly of the face, the lips, purposeless. But the thing is here, rhythmic. You know, unlike the jerky things, these are, you know, they'll do it for 10 minutes and then stop, and then start again. Blair?
What's DA stand for? It says inhibit DA action.
Dopamine.
Dopamine.
You remember the substantia nigra. The problem with Parkinson's disease from the substantia nigra is loss of dopamine projections to the striatum. There are two main projections to the striatum. One contains dopamine, which is excitatory. The other is acetylcholine, which is inhibitory. So you have to sort of, and I'm not going into this, I purposely did not go into this, but you have to think of what's going on here. Okay, so the substantia nigra projects to the caudate with dopamine and acetylcholine. So there's a balance there between excitation and inhibition, right? So if you lose dopamine, you have nothing but inhibition. So now you inhibit the caudate's projections to the globus pallidus, allowing the globus pallidus to fire. The globus pallidus fires, it projects one place to the thalamus. Remember, the globus pallidus is GABA, so if it fires unusually a lot, it's going to inhibit the thalamus. The thalamus projects to the cortex. The thalamus modulates the cortex. If you now inhibit the thalamus, it allows the cortex to set free. So bam, you got movement. So I didn't want to get into that, but that's the thinking behind this, the physiology of it, okay? And I condensed it all down to one statement: disinhibition. Release phenomenon. That's all you need to pay attention to. Okay? So here we go. In some different types of movement disorders. We know about Parkinson's and Huntington's. There's another one called Sydenham's chorea. Is what you see in children. It can happen in untreated strep infections, and what they are is tiny, tiny, tiny little hemorrhagic lesions, and particularly the, I believe it's the subthalamic nucleus. I think it's subthalamic. That and hemembolism, I think both are subthalamic. The thing about Sydenham's chorea is that it's self-limiting. It goes away after weeks to months. But it's a pretty significant, large dyskinesia. It actually has the name, the old name for it was St. Vitus's dance, because these little kids would be just flailing along here. I have seen one case of Sydenham's chorea in my career, but you know, you treat strep throat now, so you don't really, it's kind of gone away. do a YouTube of St. Vitus's dance, and you'll see what this looks like. Hemibolism is a similar thing. It's a lesion, small strokes in the subthalamic area, and it produces wild flailing type movements, one side and contralateral to the lesion. These also are self-limiting. They will usually go away over time. Finally, Wilson's disease is also called hepatolenticular, oh, there it is right there, hepatolenticular degeneration, because what this is your inability to metabolize copper. And copper accumulates in the lenticular nucleus, which is part of the basal ganglia. You know, I said the different nuclei groupings have different names. One of them is like striatum, the lenticular nucleus. So you start damaging the basal ganglia. It also accumulates in the liver and produces a cirrhosis. They build up a copper. Anyway, it's a flapping like this. You get them to hold their hands out and they flap like that. It's not a wild flailing thing. It's the goofiest look. If you YouTube image it, it's a pretty interesting thing. One of the pathognomonic hallmarks of Wilson's disease, which I've never seen because it's diagnosed at an early age and treated, is the Kaiser-Fleischer rings, which is the accumulation of copper in the iris, around the iris. This is a board question. And when I say this is a board question, that doesn't necessarily mean this is a test question you're going to see, but you need to know it. Okay. Any questions on the basal ganglia? That's a real brief trip through the basal ganglia. Damage to the basal ganglia is not a good thing. You know, you can get these lesions, these tremors, with not only the lesions we've described, like Parkinson's and Huntington's, but with tumors and infarcts and so on. Let's move to the internal capsule. That structure between the basal ganglia, or the caudate putamen, the putamen and globus pallidus, and the thalamus. So you get all these descending and ascending fibers from the cortex, from all 5 cortical areas. They go right through this little area right there, and then descending. This is showing the corticospinal tract, because it shows the pyramidal decussation right there. Right? If you look at this dissected view, it looks like that. That's pretty impressive, that all of these millions of fibers converge at that one little place right there in the deep brain. If you take a section through here, it looks like that. Oh, I'm sorry. It looks like that. So there's your putamen. There's the globus pallidus. There's the caudate. There's the thalamus. There's the third ventricle. This is the internal capsule. All right, you see where it says capital F-A-L and little f-a-l? That's face, arms, legs. The capital ones are the motor fibers. The lowercase f-a-l are the sensory fibers. So the motor fibers are projecting out as the corticospinal tract. The sensory fibers are the ones going up from the thalamic projections as the dorsal column or spinothalamic projections from the thalamus up to the postcentral gyrus. That's where they travel. Now here's the thing, Katie.
I think I might just be understanding it wrong. If these are, the motor fibers are going through the corticospinal tract, then why are they considered extra pyramidal?
No, these are, no, they're not.
Oh, okay. All of these are intra.
That right there is the pyramidal tract. Yes. The extrapyramidal is basal ganglia. this is parameter,
Okay.
okay? All right. So here's the point I made the other day, and I said, when I get to this point, I want you to remind me to focus on it for a second. If you look at the arterial supply to these deep brain structures, here's the middle cerebral artery. And right there, you see those little arteries? Those are the striate arteries. I drew a Frank Netter-like picture on the board of these striate arteries the other day. You remember that. I'm sure you all photographed it. So those are the striate arteries right there. Those striate arteries project to the internal capsule. So one little artery supplies one little area of the internal capsule. This is the point I want to make here. If you have a stroke of the middle cerebral artery, then it usually involves this, which gets both motor and sensory components. So a stroke of the middle cerebral artery, they will have motor and sensory deficits. The striate arteries supply only a small area. Each little striate artery supplies only a small area of the internal capsule. So if you blow a striate artery, usually as a result of hypertension, as I mentioned earlier, you will have a sensory or a motor deficit, but usually not both. Because the face is separated, the facial fibers, the sensory facial fibers are separated by a pretty good distance from the motor facial fibers. Same with the arms and legs. You have a motor or a sensory deficit, but not both. I mentioned before, this is called a lacunar implant. Here's another picture of the striate arteries. They're beautiful little arteries, but they're tiny, tiny. All right. Back in 1909, this cowboy named Brodmann, I think Leroy was his first name. Not really. I don't know what his first name was, but Brodmann was his last name. He identified 40-something areas of the cerebral cortex based on the cytoarchitecture. So areas that all look the same in terms of the layering of cells, the size of the cells, all sorts of things like that. He gave them a specific number. And so these are known as Brodmann's areas, and these are still used today. And there are a lot of them. We're going to cover some of them here that you'll, today and tomorrow, so here we go. We already know that the precentral gyrus is here. That's the primary motor strip. That's area 4. The postcentral gyrus is the primary sensory strip. The reason I remember that motor and sensory is because if you're going from front to back, M comes before S. That's how I always remember that. The sensory strip is area 312. You call it 3, I don't know why you call it 312 instead of 123, but that's what you do, 312. We already know those. Okay? Talk about thalamus. Here is our sagittal section. That large bundle of fibers there, we'll talk about tomorrow. That's called the corpus callosum. It's the main connection between the right and left hemispheres of your brain. The corpus callosum. Right under the corpus callosum, you'll see a little band of fibers right there. That's called the fornix. We'll cover the fornix. I'll mention the fornix here in a little bit. That band of fibers right there. Right below that is the thalamus. So you have two thalami. Between them is the third ventricle. So this is the third ventricle opened up here. We've cut, right through the third ventricle. connecting the two thalami in a subgroup of people, not everybody has one, is this connection right here called the massa intermedia. It's a non-neural connection. I don't even know why it's there. But it's not there on everybody, but you'll see it on an MRI on occasion. It just connects the right and left thalami through, it's a bridge across the third ventricle for some reason. It's non-neural. You can see it in this illustration right there. So this is where we're targeting, right there, the thalamus. The thalamus looks like, it's shaped like a football. You have two footballs deep in your brain. There it is right there. Again, to remind you, it's the lateral wall of the third ventricle. The thalamus is divided into several different nuclear groups. And here they are. I want to cover a couple of these nuclear groups. We already know what the VPL is. What information goes to the VPL?
The trigeminal, no.
Not the trigeminal,
Sensory.
that's the VPM.
Oh my
The VPL receives sensory input from the dorsal column and the spinothalamic pathways. This is where the third order neurons are that are going to project to the postcentral gyrus. The VPL, ventral posterior lateral. Here are two little structures we're going to talk about, I think today. The lateral geniculate is a relay for vision. The medial geniculate is a relay for hearing. The dorsomedial group, or nucleus, projects to the frontal cortex, or it receives projections from the frontal cortex, I should say. And the anterior nuclear group, right here, receives projections from the mammillary bodies. Remember the mammillary bodies on the ventral brainstem thing we saw earlier? Yeah. You're going to see them again. Here's the subthalamic nucleus I had talked about earlier, in case you want to know where it is. And there are other, yes, Claire?
Which one did you say receives projections from the frontal cortex?
The dorsomedial.
Okay, thank you.
There's another nuclear group I may mention later on called the centromedian nucleus, the CM, centromedian nucleus. These are the projections to and the projections from the afferents and the efferents from these different nuclear groups. You see the VPL right there? There's the precentral gyrus, the postcentral gyrus. You can see it better on this illustration. Pre and postcentral gyrus match the colors with the color of this. Voila. Okay? Frontal lobe. Let's see here. Where it says prefrontal cortex up there, receives projections from amygdala and temporal cortex. I said earlier it receives projections. It projects to the prefrontal cortex. I apologize for that. The dorsomedial group. One thing about the dorsomedial group, have you guys ever watched One Flew Over the Cuckoo's Nest? Mm-hmm. Right? You know about the prefrontal lobotomies, people who were criminally insane. They would actually go in through the nose and go up into the anterior cranial fossa with sort of an end-to-egg egg scrambler, and just scramble the prefrontal cortex right out here. The prefrontal cortex, if you lesion it, it causes you to become docile. No motivation, no argument, you are just docile. This was a common procedure done up into the 60s or so for people who were just insane. Particularly those criminally insane, because there were no pharmacologic management at the time. John Kennedy's sister had a prefrontal lobotomy. Our former president, if you don't know who he is. So, okay? You can achieve the same thing, the docility, by lesioning both dorsomedial nuclei. Also, if you do a prefrontal cortex, the dorsomedial nucleus shrivels up. So it's important in behavior. All right. I put a couple of these slides in here. You can follow the color patterns if you want to. The lateral nucleus, lateral genicular nucleus, is the target for retinal projections. So in anatomy, we talked about the retina. There's the chiasm. There's the optic tract. The optic tract ends in the lateral geniculate body of the thalamus. The lateral geniculate, the nerves of the lateral geniculate project back to the visual cortex in the occipital lobe. These are called this is called the optic radiation. And it lands primarily in area 17. If we go to Brodmann's, there's the lateral geniculate right there. It projects to area 17. If we go to Brodmann's area, wherever that was, there's area 17. It says it's the principal visual cortex, the primary visual cortex. The primary visual cortex then takes that image and sends it to the association visual cortex, which is right beside it. That's areas 18 and 19, visual association areas. This is the cool thing. I'm going to tell you the cool thing. You ready for the cool thing? Everybody follow me so far. The lateral vision projects directly through the lateral geniculate to area 17. Area 17 sends it to areas 18, 19. If you lesion area 17, you lose the primary area. What did I say about lesioning the primary area? If you lose that, you lose the function. So if you lose area 17, you're blind. If you lose area 18 and 19, but 17 is intact, you can still see, you just can't interpret what you see. For example, if I lose area 18 and 19, and I'm looking at you and I'm thinking, I know what you are. You're a human. But I don't know, I don't know that you're a girl. I can't interpret what I see. I can see grandma, but I don't know she's grandma. That's the difference between primary and associational area. Miss Sears.
I think I've seen a movie about that where the lady like hits her head and I think she lesions that and she can't recognize people anymore and she can't like recognize her husband or anything, so she puts a different tie on him every day so she can like pick the tie or something. Would she not be able to recognize that either?
You know, I don't know. I haven't seen that movie, but sounds kind of interesting. But bottom line, the association areas, there are many association areas. They interpret what is sent there from the primary function. Okay. The other thing we need to pay attention to here is, remember in anatomy, we talked about these crossing fibers and bitemporal hemianopsia, right homonymous hemianopsia, left, that crap's coming back here, folks. Understand that this lateral geniculate, the left, we'll call this the left lateral geniculate. If you follow the pathway here, sees or receives information from the right nasal retina that sees the right temporal visual field. The other half of it projects or receives projections from the left temporal retina that sees the left nasal visual field. So in other words, the left geniculate sees the right side of your environment. It's just crossed. Which means that the left occipital cortex sees the right side of your environment. It's easy. You just follow the little colors here. It's easy. Your whole brain, if you don't remember, when you pull the brain out, it's all color coded. Okay, any questions about that?
I have a comment that's a little off topic, but I think it's really cool. Have you heard of the disease where people will get, not diseases, but people will get trauma to, I actually think it's part of their temporal lobe, but it's involved in a projection, and they'll completely ignore the side opposite of that. So people come down in like full makeup, full everything, on just their right side of their body, and it's like they don't even know their left exists.
It's called hemi-neglect.
It's crazy.
And it can happen in several places. The optic radiations go through the temporal lobe to get back to the occipital lobe. So any lesion here, any lesion of the geniculate, remember also the retinal fibers project to the superior colliculus. I mentioned yesterday, the superior colliculus forms the tectospinal tract, which is the thing I was talking about, the visual reflex pathway where you turn your head. If you lesion the superior colliculus, which also sees the other side of the body or environment, if you lesion the superior colliculus or the projections here, you have hemi-neglect. So if you get somebody, and we'll kind of talk about this a little bit tomorrow, if you get them to draw a clock face, they'll do something like that. Or they'll come in having shaved only one side of their face. But yeah, it's common in lesions of the visual pathway. All right. These, I mentioned this to you before, a long time ago. These are called circumventricular organs. They're areas where ordinarily you have a cerebrospinal fluid brain barrier. The ependymal cells that we talked about, the glial cells, the ependymal cells, they line the ventricles, except in these places here. So these are places where whatever's going on in the cerebrospinal fluid can gain access to the brain. One of these is thought to be involved, if you stimulate area postrema here, you throw up. It's known as the vomiting center. The pineal gland sits at the back of the third ventricle. It's a midline structure. The pineal gland I'll talk about that in just a second. The other one sitting at the bottom of the third ventricle here is the posterior pituitary. These are all called circumventricular organs. And basically they're defined as places where the cerebrospinal fluid brain barrier doesn't exist. If you look at the pineal gland in particular, in humans, it's not that big a deal. Maybe. In lower vertebrates, it certainly is, because a lot of circadian rhythms originate here or are influenced here. The pineal, let's see here. The pineal receives projections indirectly from the retina, which is viewing light versus dark. You got to think evolutionarily, when you were living in a cave or running around in sub-Saharan Africa, there was before clocks were invented, there was a daylight time and a night time. And that varies throughout the year. So if you think of something like a deer or an antelope or lion or something like that, reproduction, you want to have your babies when the climate is best for them to survive. You don't want to be having babies in the middle of the winter. You want to get pregnant in the middle of the winter, so you can have babies in the spring and summer. So the pineal gland in lower animals helps regulate ovulation. That's putting it very simplistically, but that's one of the factors that regulates ovulation, based on the daylight cycle. In us, it's not so obvious unless there's a problem. And you see here, if you have a tumor of the pineal gland, and the pineal gland has a particular neuron in it called a pinealocyte, in case you're on Jeopardy. If there's a tumor there and the output of the pineal is increased, it delays the onset of puberty. If the pineal is lesioned, you wind up with precocious puberty. In other words, development of secondary sex characteristics, breast buds, facial hair, that sort of thing, at the age of 8. Now, I want to caution you about something. Depending on who you read, you might get an exactly opposite response here, but we're going to stick with this, okay? We're going to stick with this interpretation. All right. Take a break and we'll come back.